Welcome everyone. I am Ellen Gualtieri, the FRAP and Rock Imager Product Manager here at Formulatrix. We are very pleased to have Vadim Cherezov from the University of Southern California online with us today to review some of the fundamentals of FRAP and to also discuss data interpretation and understanding. We have over 60 attendees today with a mix of current and potential users, so we're hoping for some great discussions and questions at the end. It looks like we are now ready to begin, so we'll turn the presentation over to Vadim and get started. Thank you.
Hello everyone. My name is Vadim Cherezov. I'm a professor at the University of Southern California. And today's webinar I will be giving you bolts and nuts of using LCP FRAP assay for screening membrane protein crystallization in lipidic cubic phase. This is a fairly broad topic, and we have a limited time, so for the purpose of this webinar, I assume that majority of the listeners know what lipidic cubic phase is, and they use this crystallization technique for crystallizing the membrane proteins in their lab. And I also assume that many of you have the LCP FRAP instrument and already started to use it. I also hope that at least a few of you have read our original publications on the development of the LCP FRAP assay shown here, and also a couple of follow-up reviews and protocols, one of which was just recently published in Methods of Enzymology. The outline of today's webinar is as follows. I will start with a brief introduction into the lipidic cubic phase crystallization and diffusion of membrane proteins in lipidic cubic phase, and on the use of fluorescence recovery after photobleaching for looking at the diffusion of the protein. I'll then go into protein labeling and give you some more details about sample preparation, because this is one of the most important parameters for the success of this technique. And most of the experience during our development and the use of the LCP FRAP assay related to the use with G-protein coupled receptors. However, there is I don't see any reason why this method cannot be used for other membrane protein families. I'll then give you several examples of using LCP FRAP assays for guiding crystallization trials. And then I will switch to troubleshooting section and cover several of the common problem and mistake that user experience during the use of these assays. And I will finish my webinar with the question and answer session, when I will answer some of the common questions that our users will submit for this webinar. As many of you know, lipidic cubic phase can be prepared from special lipids such as monoacylglycerol monoline, by mixing it with about equal amount of water. Upon mixing, lipid will assemble into lipid bilayer that resemble the environment of biological membrane, and this lipid bilayer folds in three-dimensional space to form these three-dimensional periodical structures with cubic symmetry known as lipidic cubic phase. There are two important facts about lipidic cubic phase. First of all, that it provides a good mimic for the environment of biological membranes. Membrane protein can be inserted into these lipid bilayers. And the second, that it has a bicontinuous properties in such that both hydrophobic and hydrophilic molecules can diffuse continuously in three-dimensional space inside of this matrix. It consists of a single lipid bilayer that spans this three-dimensional space, divided it into two networks of water channels. And there's a continuity in both lipid membrane and water channels. One can insert membrane proteins into the lipid bilayer, and it should be able to diffuse in three-dimensional space following this lipid membrane. And components of the precipitate solution can diffuse through water channels they can change the properties of lipidic cubic phase and also modify interaction between proteins, inducing nucleation and crystal growth. In order for crystal to grow, it requires that the protein should be able to diffuse freely in the lipidic cubic phase. But due to this specific microstructure and spatial constraints, lipidic cubic phase can impose certain limitation on diffusion of large membrane proteins or large soluble molecules. And therefore it's important to understand and study the diffusion of membrane proteins in the lipidic cubic phase in order to understand and predict how crystallization occurs. So let's look a little bit more in details of how membrane proteins diffuse in the lipidic cubic phase. And here I'm showing you in panel A a single knot of lipidic cubic phase with p-n-3n symmetry. As you can see, not all of the points on the surface of the membrane are equivalent. There are points such as those that marked with a star, which has the lowest absolute curvature. This point are known as monkey saddle. So the proteins are most happier in these points of the lowest curvature. So they sit in these monkey saddle points. In order to move from one monkey saddle point to another, they need to pass through a region with the highest absolute curvature, such as that shown in panel B. So in that point, the free energy is the highest. And depending on the size of the protein and the curvature of the lipidic cubic phase, so the barrier could be sufficiently large to substantially limit or even completely halt diffusion of membrane proteins. Also, I can imagine that membrane proteins, several membrane proteins can assemble together at the monkey saddle point, and then they can oligomerize in that point, forming a sufficiently large aggregate that would not be able to pass through the high curvature regions. And again, the diffusion of such proteins will be very limited. One of the common ways of looking at the diffusion is fluorescence recovery after photobleaching. The technique is very simple. One needs to label membrane protein and insert it into lipidic cubic phase, and then bleach a spot using a laser and look at the recovery of fluorescence in the bleached spot. The fluorescence can change only due to the bleached proteins moving outside of the bleached spot and non-bleached protein moving inside. Therefore, by analyzing the profile of the recovery curve, one can determine the diffusion coefficient of the protein and the mobile fraction. Diffusion of membrane proteins in the lipidic cubic phase is about 2-3 orders of magnitude lower than diffusion in aqueous solution. Therefore, it requires at least about 20 minutes to record the full fluorescence recovery curve from which you can deduce accurately diffusion rate and mobile fraction. We wanted to look at many different conditions, many different precipitant conditions. Therefore, we designed a so-called high-throughput FRAP technique in which we can set up 96 well plate using 96 different precipitant conditions and bleach all 96 samples sequentially, one by one, then incubate the plate for a certain period of time, and we selected 30 minutes as the appropriate time for incubation, and then go back and look at the fluorescence recovery after 30 minutes. So what we have in this case is the percentage of the fluorescence recovery after 30 minutes, which in case of the fast diffusing proteins would be equivalent to the mobile fraction, and in case of the slow diffusing protein, it would be smaller than the total mobile fraction. However, this parameter gives us a good indication on how membrane proteins diffuse in the lipidic cubic phase, and that gives us a good guidance in order to either we should follow up this condition or not. Typically, we display results of the high-throughput FRAP in this color-coded scheme, in such that the conditions in which protein does not diffuse shown as blue, conditions in which protein diffuse well shows in red, and the conditions in which diffusion of protein is limited, restricted, is shown as green. It takes less than 2 hours to collect data on 96 well plate this way. And we can also look at the full recovery curve in some selected wells after we've done the high throughput FRAP in order to more closely look at how fast the protein diffuse and determine the diffusion coefficient. Protein labeling is very important for the success of LCP FRAP assay, and I cannot sort of emphasize more that the knowledge of your protein is also very important in order to select the proper way of labeling your protein. And I will give you some guidance that you could use in order to select the way of how you should label your protein. There are 2 common ways of labeling proteins. One of them is to label free amine groups, such as free amines on the solvent exposed lysine residues and on the free N-terminus. There are several dye conjugates that can be used for this purpose. The second way is to label free thiol groups, such as those on free cysteines, and also there are a couple of dye conjugates, such as iodocetamides and maleimides, that could be used to label free exposed cysteines. There are advantages and disadvantages to both of these labeling techniques. Labeling amine groups is a universal. There are always free amines on all the proteins. We prefer to label amine groups at pH close to pH 7, 7.2, at which efficiency of labeling of free lysines is pretty low, and we predominantly label free N-terminus. So this, what we found, is the most gentle way of labeling protein, and it's the least disturbing, and so does not interfere with protein-protein interactions and crystallization. The disadvantage of free amine labeling is that lipids that co-purified with the protein that contain free amines, such as phosphatidylamines, phosphatidylethanol can be also labeled, and they end up in the final sample and produce a certain background which needs to be taken into account. And I'll show you some of the examples of this. Labeling of free thiols is more specific. You know what exactly cysteines you're labeling, but that requires the availability of free exposed cysteines in your protein. If your protein is recombinant, and there's no free exposed cysteines, you can perhaps engineer a cysteine in an unstructured part of the protein that will also not interfere with protein mobility and crystallization. What we found important is to label cysteines when the protein still sits on the membrane, because very often cysteines in membrane proteins occur at the transmembrane helices inside of the membrane part, but close to the surface. So these parts are usually shielded from the dye when the protein in the membrane, sitting in the membrane, but when it's solubilized in detergent and becomes more dynamic, they can be accessible to the dye and be labeled, and insertion of a bulky dye at this position can be destabilizing for the protein. And again, the good point about cysteine labeling is the lipids are not labeled. For the purpose of labeling GPCRs, after long trial and error, we end up on using a size 3 dye succinyl ester and label them at pH around 7.2 to predominantly label free N-terminus. So that's what we found works for the most of the G protein coupled receptors. There are several strategies to label a protein. One of them is to label protein while it still sits in the isolated membranes, and this way is recommended for cysteine labeling. However, that requires a large amount of dye because there are competition with other membrane proteins that are present at this point. After labeling, you can just follow up your already established and optimized protocol for protein solubilization and purification. And during this process, free dye will be removed as all other impurities. The other strategy is to label already solubilized and purified protein. This is a fairly straightforward. You just combine solubilized protein with a dye in the dark at 4 degrees, incubate for a few hours, and then you need to completely remove unreactive dye by preferably immobilizing protein on an affinity chromatography resin and washing out the free dye. If your protein doesn't have any tags or ligands to be attached on affinity resin, you could use several rounds of desalting or dialysis or size exclusion purification. But you really need to be sure, make sure that all of the dye, free dye, is removed from the sample. And the last strategy is sort of a modification of the second, when you label your protein during the purification. So when the protein is bound, for example, on the affinity chromatography resin, you can label and purify at the same time. Regarding the labeling ratio, if your protein has multiple labeling sites, it is important to label it at a fairly low labeling ratio, 5% or less, so that you do not label two sites on the same protein. And we used labeling ratio between 0.1 and 5% successfully, depending on the final concentration of the protein. Regarding the protein concentration, you can also use quite a wide range of concentration. We use from 0.5 to 50 milligram per milliliter, and the considerations here as follows. In the beginning, when your protein may be quite unstable, you don't have it in large quantities, or you cannot concentrate it to high concentration, you can use a relatively low concentration of protein, such as 0.5 or 1 milligram per milliliter, and still get a reasonable idea of how your protein diffuses in the lipidic phase. When your protein becomes more stable, you can concentrate it, or you have a fairly large quantities of the protein. It's preferable to concentrate it to at least 10 milligram per mil or higher. And in that case, you will be doing your FRAP experiment at conditions that closely resembling the crystallization conditions. And another benefit is that you might detect some initial very tiny crystallization hits already in the LCP FRAP assays. And of course, since your protein is labeled, this detection is very easy using fluorescence image. Another question that often comes up is what plates users should use to set up LCP FRAP assays. And you certainly need to use so-called glass sandwich plates, which were developed about 12 years ago, and now they are available from many different vendors in different configurations. And there are different names that are associated with that. And these plates are a little bit different in the way that some of them have, for example, this plastic frame. They might use different glass coating, but one of the most important parameters in consideration of using for FRAP assay is the thickness of the spacer. And the idea here is the following, that the bleaching depth of the laser depends on the property of the laser and also on the numerical aperture of the objective used in the instrument. For the LCP FRAP instrument produced by Formulatics, the depth of field is about 50-60 microns. So this FRAP sandwich set was specifically designed to match these depths of field for the laser so that it will bleach through the whole sample. In that case, the diffusion can be reduced to two-dimensional diffusion, and the equations to analyze this diffusion is the most straightforward, and you can get, by fitting with them, you can get accurate values of the diffusion coefficient. You can also use 100 micron, 140 micron spacers, the other plates that you typically use for crystallization, In that case, not all the samples will be bleached, but you still can get data that you can use to guide your crystallization trials. However, you might not get very accurate value for diffusion coefficient. One of the drawbacks of using FRAP sandwich plate is that the glue that is used for the spacer in this plate has low tackiness. So you need to apply fairly large pressure to seal these plates. If you apply this pressure on top of the glass over the sample, you might actually disintegrate or destroy your sample. I'll show you some examples. So you need to use some special techniques for sealing these plates, and I advise using a glass or wooden stick and press in between the well and also at the edges of the top glass slide in order to properly seal these plates. When we first started to use LCP FRAP to look at the diffusion of different size protein in different conditions, we found that indeed diffusion strongly depends on both size of the protein and condition, and very often in quite unpredictable ways. We found that, for example, many GPCRs do not diffuse in many conditions at all. When we look at conditions close to the crystallization conditions, for example, here we look at the beta-cholinergic receptor at different concentration of precipitin components, we can see a good correlation between a high diffusion rate and high mobile fraction and crystallization window, which is shown here in gray boxes. So these are concentration at which protein crystallizes. We also found that the salt identity is very important in order to, that would determine whether protein will diffuse or not. And we used 48 salts, different salts from Hampton stock option to create a screen using two different concentrations of the salts, 0.1 and 0.4 molar. As well as 30% TEC400 and different pHs. And look at the diffusion of adenosine A2A receptor in these screens. What we found that many conditions the protein didn't diffuse at all. What you see here is the background about 10–15% that is diffusion of labeled lipids that co-purified with the protein. There were only about a dozen conditions that showed appreciable diffusion of the receptor. We took these 12 salts and we created optimization grid screens by changing concentration of salt and TEC400 and pH. And out of these 12 optimization screens, we obtained crystals of this receptor in 9 of them. So it was a good outcome of using this LCP FRAP for guiding and selecting proper salt conditions that would eventually lead to crystallization. So at this point we decided that this could be a good pre-crystallization assay. We designed a first prototype of this LCP FRAP at the lab, which could do a high throughput FRAP automatically, and then Formulatrix took it over and optimized it and created this FRAP imager, which is more user-friendly and faster. When you use high-throughput FRAP, you need to understand the levels of accuracy and reproducibility in these experiments. And to evaluate them, we perform the following experiments. We use beta-cholinergic receptor fused to T4 lysozyme, and we set up 2 plates with using 2 conditions. One is positive control conditions where we know that the protein diffuses well. And the second plate was with negative control, so that conditions in which protein doesn't diffuse. As the negative control, we chosen, in this case, 0.5 molar sodium chloride, because this condition doesn't affect the protein, but it shrinks LCP and restricts diffusion of the receptor that fused to the T4 lysozyme. And by using full FRAP, we confirmed that in case of 0.5 molar sodium chloride, we only see diffusion of lipids. This is a very fast diffusion that saturates very fast, and then there is no diffusion over a long period of time. And in the condition of the good diffusion, you can see protein diffusion. So when we set up 96 conditions with both of these conditions, you can then evaluate what the accuracy and reproducibility you can get. And what's fairly good, about 4–5% variation across the plate. And so this experiment can be used to set up a threshold. For example, in this case, I would set up a threshold about 20%, above which I would be certain that the protein is diffusing. If the recovery is between 15 and 20%, there might be some diffusion, but it will be fairly limited. If the recovery is less than 15%, most likely protein does not diffuse. In order to look at further correlation between diffusion and crystallization, we set up we used beta-turbonergic receptor labeled and concentrated to 50 milligram per ml and set up FRAP screens. And then we performed a FRAP experiment, a high-throughput FRAP experiment after 12 hours incubation. And then we waited for another 7 days and looked at the fluorescent images for appearance of crystals. And then we scored crystals by using a score from 0 to 4, where 0 was no crystals, 1 was a shower of very tiny crystals, and 2, 3, and 4 was crystals with increasing size. And the threshold that we used in this experiment was 28%, so below which protein didn't diffuse, and above which there was some protein diffusion. And as you can see, there are many conditions in which protein didn't diffuse at all, and there was no crystals. There were 6 conditions in which there was protein diffusion but no crystals formed. And that is understandable. Diffusion is required but is not sufficient for crystallization to occur, because you need to satisfy other requirements for protein-protein interactions in order to crystal nucleate and grow. There were many conditions in which we saw diffusion and crystals, and also the crystals appeared. There were, however, 4 conditions in which no diffusion was recorded, but we found very tiny crystal hits in these samples. When we look back at the images recorded at the time of the FRAP experiments, 12 hours past setup, we observed that in these cases the crystals have already formed by the time we performed a high-throughput FRAP experiment. Therefore, protein actually already either crystallized or aggregated by this time and didn't diffuse anymore. Crystal didn't grow past this point. So overall, it was a very good correlation between diffusion and crystallization in this experiment. So after that, we decided that we would use these assays for any new protein target, and we use it in different ways to screen for precipitant conditions and select most promising conditions for the further optimization screens and remove those salts that are not conducive to the protein diffusion. We screen for different ligands. We screen for host and additive lipids, and we also screen for different protein constructs. And I'll show you further a few examples of using LCP FRAP assays for screening, for guiding crystallization trials of G-protein coupled receptors. The first example is a fairly old one of a CXCR4 chemokine receptor. So this receptor was particularly difficult and it didn't diffuse well in the lipidic phase. After many rounds of optimization of the protein construct, we end up at these constructs that had some diffusion, fairly low, about 30% in 4 conditions. So we took conditions that with the best diffusion, sodium malonate, and set up optimization grid screens by changing concentration of PEG 400 and sodium malonate. And we observed a very tiny crystal hits in this screen. By further optimization using additives, we got little beta crystals, and further optimization of all concentrations, we obtained final crystals that were sufficient size to get a high resolution diffraction synchrotron source. The second example is for looking at different subtypes of a serotonin receptor. And these subtypes have a very high similarity, and they bind to the same ligand. So we prepared exactly the same construct for these 4 different subtypes and looked at them with the same ligand. And what we found that although the property of these proteins were exactly the same in solution, they behave very different in the lipidic phase, with 5-HT1B protein diffusing the best and other subtypes didn't diffuse almost at all. When we tried crystallization, we obtained crystals of the 5-HT1B and we didn't get crystals in any other subtypes after extensive crystallization trials. The next example is for looking at different ligands, and this is for mGlu1 receptor. It's a receptor class C. Again, the choice of the ligand is very important for crystallization and also for diffusion of the protein. And it's often not possible to predict which ligand can actually be successful for crystallization. Just affinity is not a sufficient parameter. Also, we looked at stabilizing property of the ligand in solution, but that's also not the whole story. Well, we found that the diffusion of the protein LCP is a better metrics for selecting the best ligand. And in this example, we looked at 2 very similar affinity ligands, which have similar stabilizing property for the protein in solution. And again, in one case, ligand, the protein complex with ligand shows a good diffusion, while in the other, diffusion was very poor. And again, the protein that showed the good diffusion, that was the one complex that was crystallized, and the structure of which we have obtained. Also, diffusion of the protein is related to stability, and you can look at diffusion results in different points of time to see how stable your protein is in different conditions. And also use this to understand for how long you actually need to follow up your crystallization trials. For example, in this case, for sodium sulfate, diffusion of protein was decaying fast in time, and after 7 days, there was almost no diffusion, and crystals didn't grow past 7 days. Whether in sodium citrate, there was still a pretty high diffusion even after 7 days, and crystals continue to grow even after 7 days. So this is a good way of looking at protein stability in the lipidic phase as well. And as I mentioned, the beneficial point of labeling the protein for LCP FRAP assay that you can actually very often spot initial crystallization hit right in the LCP FRAP assays. As shown in this example, these tiny crystals cannot be detected by using bright field image or any other imaging techniques. And of course, if you get your initial hit, that's your good point for further optimization. Now I'm going to troubleshooting side section, where I will show you 4 main problems that users often encounter when they set up LCP FRAP assay or analyze the data. The first set of problems related with dispensing LCP and plates sealing. When you set up your sample plate, you need to make sure that your LCP bolus has this round shape and sandwiched fully well between 2 glass slides. Very often, if you press too hard during the plate sealing, you can create some indentations in your sample or in completely disintegrate LCP bolus. If your LCP needle of the robot comes too close to the plate or even touches the plate, you also get some irregularity in the dispensing of LCP. If your precipitant tip can come too low, you also can create a footprint on the LCP droplet. And I cannot even speculate what happened in the last case where this Mickey Mouse head shaped drop was dispensed, but this is also a very common occurrence in the dispensed drops. So in all of these cases, this irregularity in the shape should be noted, and the reason is that the diffusion of the conditions in the lipidic phase depends especially in the beginning, in the first few hours when the equilibration happens, they depend on the shape of the lipidic cubic phase balls. Precipitant can only diffuse from the side in the sandwich plates, and it creates gradients of precipitant concentration that can affect behavior of the protein in LCP balls. Therefore, the shape of the droplet is very important in order to compare different conditions. If you compare conditions that have different shapes, that could not be directly comparable. Also, if you have these irregularities in the dispense drop, a bleaching spot can come into the place where the sample is disturbed or there is no sample at all, and again the results will be not reliable in that case. The second set of problems related to the setting up the exposure time, and exposures can vary dramatically in these samples because of the labeling efficiency can be different for different proteins, and also different conditions can change the fluorescence. And even within each drop, fluorescence can change dramatically due to the presence of precipitated protein and some other aggregates. Therefore, this parameter is user selectable, and users should look at several droplets and choose a proper exposure time. If your exposure time is too low, as shown in this case, in most of the droplets will get too low signal, and it will be a high error associated with these experiments. If your exposure is too high, then you get overexposed samples, and again the results will be unreliable in this case. The third set of problems related to the phase change. Some precipitant can induce phase change from lipidic cubic phase into another phase, and that often accompanied with either protein precipitation or creation of some other irregularities in the lipidic cubic phase drop. Such conditions should also be noted, and results that comes from them also could not be trusted. The last set of problem related to protein precipitation or occurrence of crystals in the droplet. And of course, if you bleach a part where the protein is precipitated or the part of the droplet where there are crystals, you might get no diffusion of the protein, while in other parts of the LCP the protein will be diffusing well. For this particular reason, there is a feature in the LCP FRAP instrument where you can bleach two different regions, one close to the middle of the drop and the second one close to the edge of the drop. So in that case you can profile the diffusion in two different locations, and that can give you a better understanding of how protein diffuse in this lipidic cubic phase drop. And finally, I would like to note that this LCP FRAP assay is designed to look at the diffusion of membrane protein in the lipidic cubic phase. However, there are several common precipitants that at high concentration can swell lipidic cubic phase and induce formation of so-called sponge phase. Sponge phase has a similar topological properties in such that it can consist of a single lipid bilayer. However, the long-range odor is lost in this case, and there are large fluctuations, and it has a liquid-like properties. Therefore, there is no more restrictions on diffusion of a large membrane protein or aggregates of the protein in sponge phase. And the results of the LCP FRAP assay would be inconclusive. You might get an aggregate protein which still show diffusion in these assays. They might be possible to distinguish between diffusion of single protein and oligomeric aggregates by looking at the very fast part of the diffusion, but that requires optimization of these assays, and it's not currently yet implemented in the current version of LCP FRAP instrument. This doesn't mean that you cannot crystallize your membrane protein in sponge phase. In fact, if your protein size is larger than 100 to 150 kilodalton, you should design your screens in order to induce sponge phase and try to crystallize it in a sponge phase, because diffusion of 100, 150 kilodalton protein, and larger will be severely limited in the proper lipidic phase. I'm now continuing to questions answer session. So these are the questions that were submitted by the viewers of this webinar. So the first question is, how critical is the temperature during the LCP preparation, and how long do you incubate it at 20 degrees before starting the experiments? And what lipid additives do you use? The temperature when you prepare LCP is important, and you would like to keep the temperature around 21 to 23 degrees Celsius. Especially when you work with monoolane-based lipidic phase, because if you have a higher than 23 degrees, so your protein will be less stable. Below 20 degrees, your protein, your LCP actually can have a chance to freeze and form a lamellar crystalline phase. So this window of 21-23 degrees is sort of ideal to set up your samples. If you work with shorter chain lipids such as 7,9, 7,7 mag, you can work even at lower temperatures than 21 degrees. Incubation typically takes a few hours for conditions to fully incubate, and we typically incubate samples overnight. Anywhere between 6 and 12 hours after the setup before we start doing FRAP experiments. And you can add most of the lipids, either native or synthesized lipids, up to about 10-15 mol%. For charged lipids, you can add up to about 5 mol%, and it's often very good strategy of adding additive lipids, especially if you know that specific lipids are required for your protein function, or some of the lipids are can stabilize your protein. For example, in case of G-protein-coupled receptors, we always add about 10% of cholesterol into the lipidic phase. They always help to stabilize and crystallize G-protein-coupled receptors. And the second question is, for a multi-subunit membrane protein or a complex between a membrane and soluble protein, how to choose which subunit protein should be labeled? And here I should again emphasize that the knowledge of your target protein is critical, and you should really understand your system and understand which part of the protein is better to label to distribute the least. It's a good strategy to try labeling the different subunits or try to label different partners in different experiments so that you can follow diffusion of each of the subunit or each of the partner, and it can get you an idea if the complex has formed or these subunits or individual proteins diffuse on their own. So that's a sort of a good strategy to try labeling different components and look at the diffusion of them in LCP. Question number 3, do you often see any false positives with LCP method? I'm asking this because there's so many false positives with the BICEL method. And so this is related not to probably LCP FRAP assay, but to crystallization in lipidic phase and false positive crystals. And indeed with BICELs, it's very common to get lots of false positive crystals of detergents or lipids. In LCP, false positive crystals are also possible. However, they're fairly rare. Mostly what you can get are crystals of salts or ligands, and they can be often easily distinguishable from proteins by using intrinsic protein fluorescence. Question number 4, does the presence of fluorescent tag affect crystallization? Well, potentially the presence of fluorescent tag can affect crystallization. And this is again, the knowledge of protein is important to understand what the best way of labeling the protein. In our experience with G-protein coupled receptors and using size 3 succinyl ester, we do not observe any interference with protein diffusion or crystallization. There were a couple of cases where I've seen effect of labeling on crystallization. That was with labeling bacteriodopsin. If you label it at N-terminus, it can interfere with crystallization because the packing is very tight and terminus and the basic attachment of the dye can disrupt the protein-protein interaction between layers. And also we've seen once a problem when we labeled cysteines on the GPCR, and in that case the protein actually crystallized, but it was only unlabeled protein that crystallized and labeled didn't. So the crystals that appeared were not fluorescent, but that was the protein. Other cases when we labeled free N-terminus with size 3 succinyl ester, we didn't see any effect on diffusion or crystallization. Question number 5. In our test experiment with bacteriodopsin, we got a labeling ratio of 20% using size 3 succinyl ester. Lipids should not be labeled in this case. Not sure what causes this high number. Without knowledge of exactly the procedure that you used, I can only speculate that the dye may not be completely removed after the labeling, and that would look as a high percentage of labeling, or the concentration of the dye was used too high, or the pH was different from what we typically use. So this labeling is very sensitive to pH. If you label at high pH, 8 or so, you can label more of the residues in the protein and the labeling percentage could be higher. Question number 6. How can one overcome the problem of the protein precipitating in the center of the bolus? So that was the example that I showed you earlier when the protein can precipitate in the middle. What you can do is decrease precipitant concentration. In that case, your dehydration in the middle of the droplet will be less and protein may not precipitate. You can also decrease the volume of the bolus. In that case, the gradients will be also lower, so the dehydration will not be as severe and protein may not precipitate. Or you can bleach a spot near to the LCP edge. In that case, you might still see diffusion of the protein. The protein will not be precipitated at that place. And the crystals may appear actually at that place as well. Question number 7. How can you differentiate between lipid versus protein diffusion? Can the mobile fraction number alone tell you if it is lipid or protein diffusing? So the mobile fraction alone that you see at the high throughput FRAP experiments cannot tell you exactly whether this protein or lipid diffusing. As I mentioned, you need to set up control negative and positive control experiment and look at the full FRAP curves to confirm that the protein is not diffusing, and in that case where you can only see that the lipid is diffusing by the full FRAP, recording on the full FRAP curve. Question number 8. A user has seen examples of conditions where crystals formed but the mobile fraction was very low. Any insight as to why this may occur? Again, as I mentioned, one of the reasons could be that by the time of the FRAP experiments, the crystals have already formed, and therefore there is no free protein to diffuse, or the bleach spot occurred exactly on the already formed crystal. So you really need to look at your fluorescent image, not only at the FRAP results when you're doing fluorescence FRAP experiments. So that will give you an idea whether you can trust the results or whether you can actually already get crystal hits. Another case where this could happen is the protein diffusion was measured in the center of the drop, where the protein might actually precipitate it, where the crystals appeared close to the edge, where the protein was still diffusing well. So again, I would advise you to look at least 2 different locations in the droplet, close to the center, close to the edge, In that case, you will get a more complete understanding of diffusion properties of the protein in your samples. Question number 9, what is the impact of the phase when it is on the border of LCP with sponge phase? As I explained, these assays are designed to be used for proper LCP droplets. They do not provide useful insight in case of a sponge phase. If your precipitant conditions are close to transforming LCP into a sponge phase, you can perhaps decrease a little bit concentration of the precipitant if necessary. So you should look at these images, and from the images you can actually see whether your droplet transformed into sponge phase or not. When transformed into sponge phase, it appears as a liquid droplet, so it will have a very smooth edges. Whether for LCP it's a gel-like phase, you will see some irregularities at the edges of the LCP balls. Question number 10, how many conditions do you typically screen? How many screens do you typically use? Typically for one experiment we set up about 3, 4, 96 well plates, just because this is a good number to analyze during the day. It takes about 2 hours, as I said, per one plate, so for 4 plates it's 8 hours, plus if you look at some of the full FRAP curves, that would take you the whole day of looking at these plates. So 3 or 2, 4, 96 well plates is sort of a typical setup that we use. Question number 11, which screens would you suggest to use? Are there any commercial FRAP screens that you would suggest, or do you prefer to do your own? For GPCRs we use screens based on PEG 400, 48 salts from Hampton stock options, and using a pH typically in the range of between 5 and 8. For other membrane proteins, you can either design your own screens, and again using these different salts could be important. You can combine them either with PEG 400 or high molecular PEG 4000, 2000, or you can combine them with some other common precipitants. You can also use commercial crystallization screens. However, many of these screens, especially those that are not designed for lipidic cubic phase crystallization, can flip lipidic cubic phase, transform it into another phase, and you need to make sure you need to look at the images and identify such cases, and do not use results from these conditions. But otherwise, yeah, you can use pretty much any commercial screen, and you can have many conditions that would be still compatible with lipidic cubic phase, and it can give you meaningful results. Or you can create your own screens. Question number 12, how do you determine which wells to do full recovery FRAP on? Typically we select, after we perform high throughput FRAP, we select several wells with high recovery fraction and look at the full fluorescence recovery, just to understand how fast the protein is diffusing. From the diffusion rate we can estimate whether the protein diffuses as a single monomer or it diffusing perhaps as an oligomer, as a dimer, or a higher order oligomer. We also select a couple of negative conditions just to look at them as the negative control. For example, especially if we know that there are lipids co-purified with the protein, from this data we can estimate what the percentage of the lipid in the sample. Question number 13. I would also like to understand how to best interpret FRAP data for new targets that have no historical data. How do you define a signal in the noise, etc.? You know, if you work with a new protein, you should, as I showed in one of the slides, set up one plate with a negative control, one with a positive control, and analyze the results to understand what kind of variation or fluctuations will happen in this assay. and that would give you an idea how you can interpret your data in the actual screen that you perform later on. So this is an important experiment to do in the beginning, and as soon as you understand better your protein, your target, then you can do many other conditions. You can screen for many other conditions. Question number 13. To which protein can you apply FRAP, and how would you apply FRAP to other proteins different from GPCRs? Membrane proteins, ion channels, nucleoreceptors, soluble proteins, etc. As I said, these FRAP assays should be applicable to any membrane protein different from GPCRs as soon as the molecular weight is below 100 or 150 kilodalton. For larger than 150 kilodalton, so far this method is not really applicable, and you need to use a sponge phase in order to crystallize it in this lipidic mesophase, such proteins. And we will be working further to optimize these assays for a sponge phase, and maybe in the future this will be also available. And yeah, please, if you use these assays for any other than GPCRs, please send me your feedback. I would be very interested to know the experience of people of using FRAP with non-GPCR target proteins. And finally, question number 15. What is the impact of high concentration of salts, high concentration of calcium chloride? So which concentration limits would you suggest to use? Typically, most salts alone will shrink LCP, so that would shrink, decrease the size of the channels and increase the curvature. When you use salts in combination with low molecular weight pH, such as pH 400, they actually swell lipidic cubic phase. And the limits that I would suggest when using salts alone limit to about 2 to 3 molar of salt. With higher than 3 molar for most of the salts, it can transform lipidic cubic phase into a hexagonal phase. In combination with pH 400, typically I would limit to about half a molar of salt concentration. Again, using a very high concentration of salt and high concentration of pH can transform your sample into a sponge phase. On this, I would like to, I think we have come to the end, and I would like to acknowledge the people who helped to develop the LCP FRAP assays, so my former colleagues at the Scripps Research Institute and currently at the University of Southern California. I would like to acknowledge Ellen Galtieri and Eric Zhao from Formulatics, with whom we worked a lot in optimization of the instrument. And I should disclose that although I have participated in the development and optimization of the instrument, I have no financial interest into the selling of these instruments. And I would like to acknowledge NIH funds that paid for the development of these assays. And thank you very much for your attention.
Thank you, Vadim. We really appreciate it, and thanks all for your attention and great questions. If you end up having further questions, feel free to email myself, Ellen. The email address is ellen@formulatrix.com. And thank you. Bye.